So the TCA cycle. We'll talk about this today, and then we'll finish up with on Thursday. So the idea here, right, is that we have produced a C3 unit from glycolysis, right, pyruvate. That pyruvate gets oxidatively decarboxylated to give us acetyl-CoA. That's a 2-carbon unit, and that 2-carbon unit then enters into the TCA cycle. And remember, the point of all of this, the point of glycolysis, the TCA cycle, is to oxidize carbon. And that's really the big role of the TCA cycle. It's to oxidize these 2 carbons that enter in. Okay? So really important things to note about this is we have 2 carbons coming in and we have 2 carbons going out, right? We produce 2 carbon dioxides. We also produce 3 equivalents of NADH, 1 equivalent of FADH2, and then 1 nucleotide triphosphate. And I stress nucleotide triphosphate because they can actually be an ATP or a GTP, depending upon what organism we're talking about. Okay, so the role of the TCA cycle is to oxidize these 2 carbons that enter. That oxidation, like the energy of that oxidation, is stored in the form of reduced cofactors NADH or FADH2, right? Remember, every time we go through an exergonic step, the hope is to store that energy in some sort of a chemical bond or some sort of a carrier molecule. In this case, the energy is stored in the form of these reduced cofactors. These reduced cofactors then take that energy into the inner membrane space, really the inner membrane of the mitochondria, and ultimately the electrons are given to oxygen. This giving electron to oxygen is a highly exergonic process, right? So we transfer the energy that's stored in these reduced cofactors into, and we release that energy by transferring those electrons to oxygen. That energy is released, gets captured by pumping protons across the inner membrane of the mitochondria, creating this very empowered proton gradient. Not just a concentration gradient, but also an electrochemical gradient, because we're going from a low positive charge on the inside in the matrix into a high positive charge outside. So we harness the energy of electron transfer by moving protons against an electrochemical gradient. That electrochemical gradient is then the stored energy that we use to produce ATP. So we move the protons back across the inner membrane, and that releases energy, which is then trapped by converting ADP into ATP. Every step along the way, think about it, everything we're doing, the point is to produce energy and trap that energy. Every step along the way. In the TCA cycle, we oxidize carbons and we trap that energy as reduced cofactors. In the inner membrane of the mitochondria, we transfer electrons from NADH or FADH2 to oxygen. That releases energy, which is trapped in the form of an electrochemical gradient. We release the energy of the electrochemical gradient and we make ATP. The energy is trapped in the bonds in ATP. ATP is storing energy, which is then used to power other things in the cell, right? Everything is about trapping and storing energy. Okay, so I've mentioned before that compartmentalization is important, okay? We know that glycolysis and gluconeogenesis both happen in the cytoplasm, right? Which is why it's so important to make sure we have strict controls on the activity, the flux in these 2 pathways, okay? In addition, in the cytoplasm, we have the pentose phosphate pathway, which produces NADPH, and it also is where nucleotide synthesis starts. We have synthesis or degradation of serine and glycine, of some amino acids. We have phospholipid synthesis. We have fatty acid synthesis. All of these things happen in the cytoplasm, okay? The mitochondria is the powerhouse of the cell, right? This is ultimately where we produce ATP. So we are now moving out of the cytoplasm and we're moving into the mitochondria for the TCA cycle and oxidative phosphorylation. The link is pyruvate. We've already seen that pyruvate can be moved into the mitochondria. Anybody remember when it moved to the mitochondria before? Nobody? Remember, pyruvate gets converted into oxaloacetate. Oxaloacetate gets converted into PEP. for gluconeogenesis. So the conversion of pyruvate to oxaloacetate happens in the matrix of the mitochondria. Okay, so here we are also moving pyruvate into the mitochondria, but this time it is getting oxidatively decarboxylated by pyruvate dehydrogenase to make acetyl-CoA. Okay, so the acetyl-CoA then in the matrix of the mitochondria enters into the TCA cycle. All of these NADHs and FADH2s are produced in the matrix, and then the rest of oxidative phosphorylation happens in enzymes embedded in the membrane of the inner membrane of the mitochondria. Okay, so just really quick, some anatomy of the mitochondria. So y'all probably heard this before, but I'll remind you. All right, so the idea is that we have two membranes. We have an outer membrane, which is highly permeable. Lots of things can pass through there. We have an inner membrane, which is incredibly selective. Very few things can transfer between the cytoplasm and the matrix of the mitochondria. Okay, this space between our two membranes is called the inner membrane space. It's very cleverly named. Okay, and then the other thing that's really important to note about the matrix is there's lots and lots and lots of these inner membranes. Okay, so we call these cristae. So they're basically like invaginations in the inner membrane, and the reason for that is it maximizes the membrane space. And why is that important? Why is it really important to maximize the overall area, like a surface area of the inner membrane of the mitochondria? Create more ATP, right? So the enzymes that are all important for the electron transport chain and oxidative phosphorylation, all of those are embedded in this inner membrane. So the more surface area, the more energy. All right, so let's talk about this now. Here's an overview, okay? So we start at the top, we have glycolysis, right? We know that glycolysis produces pyruvate. One of the states of pyruvate is being oxidatively decarboxylated by the pyruvate dehydrogenase complex to make acetyl-CoA, okay? And with the TCA cycle, there's a theme. And one of these themes is oxidative decarboxylation, okay? We've seen oxidative phosphorylation. I talked about it with GAPDH, how there's oxidative phosphorylation. We oxidize a carbon, couple that to the reduction of a cofactor, and then in glycolysis, we phosphorylate GAP. Here, we are coupling oxidation of a carbon with decarboxylation. Okay, so we go from a 3-carbon unit pyruvate to a 2-carbon unit acetate, and the acetate has it's coupled to coenzyme A through this thioester bond. Remember, the great value of thioester bonds is that when they're hydrolyzed, they produce energy, right? So it's a way of storing some energy. So this reaction, converting pyruvate to acetyl-CoA, stores energy in two ways. It stores it in the reduced cofactor NADH. It also stores it in the thioester bond that forms between acetate and coenzyme A. Acetyl-CoA, a 2-carbon unit, now enters into the TCA cycle. So this 2-carbon unit from acetyl-CoA of acetate gets conjugated, gets linked up with a 4-carbon unit of oxaloacetate. We've seen oxaloacetate before. When did we see it? Yeah, gluconeogenesis, right? We convert pyruvate to oxaloacetate, oxaloacetate to PEP. Good. Okay? So this is catalyzed by the enzyme citrate synthase. This makes citrate. Okay? Citrate is an isomerized to isocitrate. So isocitrate, still 6 carbons. One of the carbons is hanging off. And then, so the difference between citrate and isocitrate is the positioning of this alcohol group. So on citrate, the alcohol group is on carbon 3. On isocitrate, it's on carbon 2. Okay? In the next step, we have this oxidative decarboxylation of isocitrate to give us alpha ketoglutarate. Okay? So pyruvate dehydrogenase catalyzes an oxidative decarboxylation. Isocitrate dehydrogenase also catalyzes an oxidative decarboxylation. Okay, so we lose the carbons hanging off of the chain on isocitrate. We get carbon dioxide. We are oxidizing carbons, so we also get an NADH. We also reduce cofactor. This gives us this molecule alpha ketoglutarate, which then undergoes an oxidative decarboxylation, which is actually identical to the pyruvate dehydrogenase complex. This is catalyzed by an enzyme known as the alpha ketoglutarate dehydrogenase complex, functionally identical to the pyruvate dehydrogenase complex. Okay, we have a 5-carbon dicarboxylic acid in alpha ketoglutarate. We end up with a 4-carbon dicarboxylic acid with one of them linked through a thioester bond to coenzyme A. Stored energy in this thioester bond. This stored energy is used to phosphorylate either ADP or GDP to make ATP or GTP. Okay? We've seen this process before, right? So oxidizing a carbon to make a thioester, hydrolyzing that thioester to drive phosphorylation. We saw that with GAPDH in glycolysis. Okay? This now gives us a 4-carbon dicarboxylic acid, succinate. Okay, what I want you to note now is that succinate looks really, really similar to oxaloacetate. The only difference is we got an alpha ketone on oxaloacetate and not succinate. So the rest of the process is really just regenerating oxaloacetate. Okay, succinate gets oxidized to make fumarate. We go from a single bond between C2 and C3 to a double bond between C2 and C3. That then makes these carbons reactive so that we can hydrate it. Fumarase hydrates fumarate to give us malate. Malate looks almost identical to oxaloacetate, except we have an alpha hydroxy, an alpha alcohol, versus an alpha ketone. So in the next step, we oxidize that alpha alcohol to give us an alpha ketone. And then we start over. Okay? So when we talked about glycolysis, I told you that it is just beautiful in the chemistry, right? Like the chemical logic in glycolysis is just perfect. The same thing goes for the TCA cycle, but it's not quite as obvious, okay? So whereas glycolysis really focuses on trying to generate high energy phosphate compounds, the TCA cycle focuses around these alpha keto groups, okay? So pyruvate is an alpha keto compound, right? You have a carboxylic acid, and then a ketone, and then something else, alpha ketos. In the TCA cycle, there are 2 fates of alpha keto groups. One fate is oxidative decarboxylation, okay? Where we basically are losing the carbon dioxide. We are oxidizing this carbon, like this alpha carbon, to give us a carboxylic acid, and we're losing CO2. One fate is oxidative decarboxylation. The other fate is going to be using this as a reactive carbon for synthesis. Okay, that's important to keep in mind. The chemistry centers around alpha keto chemistry, which can either be used as a nucleation point for synthesis, the entry point in the TCA cycle, or it can be used for oxidative decarboxylation. Okay, there are 2 ways of making the alpha keto group. One is through oxidation of an alpha alcohol. So here we have an alpha alcohol. We oxidize it to produce 2 electrons. Those electrons are given to a redox cofactor, and we make an alpha alcohol. The other way is through dehydration. Okay, so we can dehydrate this to dehydrate the beta alcohol to give us an alpha alcohol, and really that is how we do it in glycolysis. It's just facilitated through an intermediate PEP. Okay, so thinking about the chemistry of the TCA cycle centering around these alpha keto groups, I think helps to remember what's going on. Okay, so let's look at this pathway again, focusing on our alpha keto compounds. We have pyruvate, we have oxaloacetate, and we have alpha ketoglutarate. These are the 3 core alpha keto compounds. Okay, what we'll note here, so I have one highlighted in blue and 2 highlighted in red. The 2 that are highlighted in red, their fate is oxidative decarboxylation. So pyruvate gets oxidized and decarboxylated to make acetyl-CoA. Alpha ketoglutarate gets oxidized and decarboxylated to make succinyl-CoA. oxaloacetate is the entry point for the TCA cycle. So it is then used for the synthesis step to make citrate. Okay? The two fates we talked about are shown here. One fate is synthesis, the other fate is oxidative decarboxylation. We talked about there being two ways of making our alpha keto compounds. And here we see in both cases for the TCA cycle, it's through oxidation of an alpha alcohol. To make oxaloacetate, we are oxidizing the alpha alcohol malate to produce the alpha ketone. To make alpha ketoglutarate, we are oxidizing the alpha alcohol isocitrate. Okay, so our alpha ketones are either used for synthesis to start the TCA cycle or for oxidative decarboxylation. To make them in the TCA cycle, both are made through oxidation of alpha alcohols. Okay, so you can think about the two steps leading up to this, right? So like to make isocitrate or to make malate, the point of the previous steps then is to make an alpha alcohol. Okay, we see to make malate, we need to hydrate a double bond. So fumarate gets hydrated. The double bond is basically like it makes it reactive enough to be able to hydrate it to give us our alpha alcohol. To make isocitrate, we isomerize citrate. Citrate, right, the first molecule in the TCA cycle, is a beta alcohol. We don't want a beta alcohol, we want an alpha alcohol. So we're going to isomerize it to make isocitrate. Okay, so we have really, so if you can focus on that, right, so the fate of our alpha ketones, how we make our alpha ketones, and then how we make the alpha alcohols, you have taken care of understanding most of the steps of the TCA cycle. Okay, we have one step here to make our double bond, right? To make fumarate, we oxidize succinate. So we have a single bond between C2 and C3 of succinate, which is oxidized to make the double bond, which is what allows it to be hydrated to make our alpha alcohol. Okay, succinate is made through a group transfer, right? So here we are taking our succinyl-CoA, we are hydrolyzing it, we're using that energy to make an ATP or a GTP, and we go from succinyl-CoA to succinate. Okay, and then the very last step here is basically just, it is an intermediate that is produced for the sole purpose of being able to make ATP, right? Because it would be very, very easy to oxidatively decarboxylate alpha ketoglutarate all the way to the carboxylic acid immediately, right? That'd be easy to do. But we'd miss out on the opportunity to harness some of that energy to make ATP in the same way we talked about with GAPDH, right? If you oxidize this carboxylic acid, you oxidize this all the way to the carboxylic acid, there's no way of trapping energy. So you trap that energy in the form of our thioester bond, which allows us then to harness that energy to make ATP. Okay, so focusing in on this chemistry, I really, really think helps. Okay, so we do not have time to talk about every single reaction in the TCA cycle. We just don't. And as it turns out, a lot of them are pretty straightforward. And your book talks about all of them. Okay, we're going to focus in on the ones that I'm highlighting here. Starting off today, focusing on the pyruvate dehydrogenase complex. And the thing I want to stress to you is that the PDH complex is this, so that if you understand the chemistry for oxidatively decarboxylating pyruvate, you understand the chemistry for oxidatively decarboxylating alpha ketoglutarate. If you just think about C3 of pyruvate as the R group, that same R group can be thought of as the other 3 carbons in alpha ketoglutarate. Okay, so just remember that. By learning this, you're also learning this. We're going to think about the synthesis step, the entry point of acetyl-CoA into the citric acid cycle. And then we're going to think about isocitrate dehydrogenase because the chemistry here is kind of cool. It's a decarboxylation. All right? So let's jump in here. Pyruvate decarboxylation. Okay? Same with alpha ketoglutarate decarboxylation is a multi-step process that involves this massive complex. You can actually see through a cryo-EM. Right? So you see like all these, so each of these blobs, that's a pyruvate dehydrogenase complex that ultimately involves multiple active sites. Okay, there are 3 different enzymes involved here that ultimately are responsible for converting pyruvate to acetyl-CoA. Okay, so here, right? So the idea, these 3 steps are: first, pyruvate gets decarboxylated to make this intermediate, which it appears to be cut off. So yeah, it's just showing a carbanion. So we decarboxylate it to make this carbanion, which then gets oxidized to make a carbocation, which then gets transferred onto acetyl-CoA. Okay, these are effectively the steps that happen. But in this process, we see the decarboxylation produces a carbanion. We don't like carbanions. They're not stable unless they're part of TPP. We oxidize it, forming a carbocation. Carbocations, we don't like them. They're not stable. And then that carbocation gets transferred onto coenzyme A. Okay, so each of these 3 steps are catalyzed by an enzyme because each of them produce something that's not especially great. Okay, so let's think about this. Oh, right. And here, so we are producing acetyl-CoA, not just acetate, not just acetic acid. The value of this is kind of twofold. One, we talked about before. This is a really nice way of trapping energy, right? So this hydrolysis of this thioester bond is exergonic, which means it can then drive subsequent reactions. In addition, the presence of coenzyme A gives us this nice handle, right? It gives us this nice recognition. Because if the next enzyme we're looking for acetic acid is kind of featureless. You got a carboxylic acid, whoop-de-doo. So does every fatty acid, right? So the idea here is that this gives us something that's structurally defined that the next enzyme can recognize, okay? So there are 3 active sites. We name these 3 enzymes E1, E2, E3 for simplicity. The first one is the pyruvate dehydrogenase, okay? This is where we are going to decarboxylate pyruvate, producing electrons that will then we give to a cofactor, okay? E2, dihydrolipoyl transacetylase. It's a big word, big name, but the idea is that when we decarboxylate pyruvate, we need to put it onto something. And we put it onto this cofactor, lipoic acid. Okay, so here we see lipoic acid. The third active site is this dihydrolipoyl dehydrogenase. Okay, in the active site of E2, we end up oxidizing lipoic acid. So we have this disulfide. And we need to reduce this back to the reactive thiol before the cycle is complete. Okay, so 3 active sites, each of them use a different prosthetic group, right? A different cofactor, a different coenzyme. E1 uses TPP. We've seen the chemistry of TPP. E2 uses lipoic acid. Okay, that's lipoamide is lipoic acid conjugated to a lysine through a peptide bond on that carboxylic acid. And then for E3, we're using FAD as a prosthetic group, as a coenzyme. Okay, and it's interesting here because this whole process, right? FAD, despite being an important redox cofactor, but despite being an important electron carrier, it's not actually reduced in the full catalytic cycle. Okay, we know the electrons ultimately go to NAD, right? If we flip back here, we know in this process that the fate of the electrons from the oxidation are NADH, not FADH2. So it's kind of interesting that flavin is used here, and it's used purely as an electron transport mechanism, right? So we're going to take electrons from lipoic acid, pass them through flavin to give to NAD. All right, so let's think about this. So the complex is big, right? So these spheres represent the blobs that we saw in our cryo-EM, right? So this cryo-EM, you can kind of see the blobs. Each of these blobs are represented by a circle or a sphere here, okay? On the outside, in yellow, is the E1 domain. That's the pyruvate dehydrogenase complex. That makes sense that it's on the outside because the original substrate is pyruvate, so the first place it hits is the outside. It makes sense. Okay? E2 is all the way on the inside. This is our dihydrolipoyl transacetylase. Okay? And then the green is our E3, and it's kind of on the surface as well. This hopefully also makes sense on the surface because this is where we need to react with NAD plus and release NADH. So we want this to be solvent accessible as well. And the idea is that these are all linked up through this swinging arm, this lipoamide domain of our E3 enzyme. Okay, and our lipoic acid, our lipoamide, is at the very, very end here. So the idea is it can swing between these different active sites. Now, we saw that when we talked about the pyruvate decarboxylase complex as well. Okay, so this is meant to kind of represent the whole process. Okay, so E1 is yellow, E2 is red, E3 is green. Okay, we start off the chemistry happening in E1. We have pyruvate coming in, we have carbon dioxide being released. Note here that this reaction is dependent on TPP. Remember, TPP is the cofactor that can produce a stable carbanion. So the nucleophile of TPP is going to be important here. Okay, we end up making this linkage, this covalent linkage between TPP and effectively acetic acid at this point, right? So we've lost CO2, we've decarboxylated pyruvate, and we now have a C2 unit covalently linked to TPP. In the next step, we see that we bring in this oxidized disulfide lipoic acid cofactor. Okay, so it swings into place once TPP is physically linked to our substrate. So this disulfide is broken. We end up making a covalent linkage between one of the sulfurs and that carbon, releasing TPP, which allows a covalent link between the lipoic acid and pyruvate. That then swings into our E3 active site. Okay, sorry, our E2 active site. At this point, acetyl-CoA comes in. Acetyl-CoA attacks the carbonyl, breaking that thioester bond, resulting in a reduced lipoic acid cofactor and our product, acetyl-CoA. Okay, the last step is to basically transfer electrons from this reduced lipoic acid cofactor through flavin and ultimately giving them to NAD plus to create NADH. So lipoic acid at this point is reduced. We give 2 electrons to flavin, to FAD. That results in the oxidized cofactor, which we need for the first step or the third step. The electrons are given to FAD, making FADH2. The electrons from FADH2 are then given to NAD plus to make NADH. Okay, so being able to kind of follow this map is important, right? So thinking about what's happening in each step. First, pyruvate gets decarboxylated, making a covalent linkage with TPP. Second, the covalent bond swaps between TPP and lipoic acid. Lipoic acid ends up getting reduced in this complex. Third, coenzyme A comes in. It takes the acetate, producing acetyl-CoA, and resulting in a reduced lipoic acid cofactor, which then transfers electrons through FAD, ultimately to NADH. Okay? So with that, and I think we'll stop there today. I'm going to give you the rest of the time to work through this question on your activity to try and think through each of these steps yourself, and we will pick up here on Thursday and go through this chemistry together.